Diffusion Lab, Section 4, Group 8B.
Katie Stocker,
Marie Camp,
Sam Gregoire. Introduction. Our problem was that when we looked at particle diffusion with a computerized model, the model did not take into account the way particles were interacting with the surroundings. So our research question was, what variables affects particle motion and how? The goals of our experiment were to determine how solution type affects particle motion. Our solution types were water, soap, and cornstarch. We assumed that these solutions have different viscosities, with water being the thinnest and cornstarch being the thickest. Our hypothesis was that particles of the same mass and size will show different motion in different solutions due to their interaction with the environment. Specifically, particle velocity and acceleration decrease as viscosity of the solution increases. Our main findings were that solution type affects particle motion. The particles in water showed the most motion, then soap, then cornstarch.
experimental design. Our variables to compare how different solution types affect the particle's motion were the solution type as independent variables, the particle motion as the dependent variables, and the particle size and material as our constants. We placed the solution with 2 micrometer beads on a slide. We connected the computer to the microscope camera and recorded movements of particles over 10-second time periods. We used tracking software to record data on a single particle movement by using the calibration stick to make the particle length 2 micrometers and tracking the particle using the center of mass tool over 10-second intervals. We then recorded data for position, velocity, and acceleration and repeated this with each solution, the water, soap, and cornstarch. From there, we created graphs from our position, velocity, and acceleration data. Some possible flaws were inconsistencies with timing between putting the beads on the slide and taking the video, which could have affected diffusion rates because it may have settled after some time, and the microscope or table may have been bumped, which would have affected the random motion of the particles and changed their direction of movement.
Data collection. Our 2 micrometer polystyrene beads in water, soap, and cornstarch.
models are 2 micrometer polystyrene beads in water, position versus time, velocity versus time, and acceleration versus time. In soap, position versus time, velocity versus time, and acceleration versus time. In cornstarch, position versus time, velocity versus time, and acceleration versus time. Analyzing data. We have each solution listed with their total change in position, maximum velocity, and maximum acceleration. The sample with water has the highest maximum velocity and acceleration. This means that the particles were able to move through the solution with little resistance. The sample with cornstarch had the lowest maximum velocity and acceleration. This means that the particles encountered the most resistance moving through the solution. The maximum velocity and acceleration of the soap solution falls in between water and cornstarch solutions. These results are consistent with our hypothesis that particles move slower through a more viscous solution.
Discussion. How would you describe the motion of the beads in solution? Does this match what you expected in the pre-lab questions? The motion of the beads in solution seemed random as beads moved in different directions with random bursts of speed. However, all the beads were moving together in similar ways. So based off the computerized model, this is not what we expected. The computerized model did not take into account interactions between the particles and the environment.
What trends or conclusions can you draw about different variables based on your experiment? In our experiment, we saw that particles in the water solution had the greatest velocity and acceleration, followed by soap and then cornstarch. The position, velocity, and acceleration graphs of particles in each of our solutions differed. This is due to the samples exhibiting random motion, but also due to the different solutions affecting their motion. We can conclude that solution type affects particle motion. The viscosity of the solution has an inverse relationship with speed and acceleration of the particles.
Random motion occurs when the position, velocity, and acceleration of the particle are unpredictable over a period of time. This can be referred to as the random walk of particles.
Diffusion is caused by collisions between particles. When there are more particles in an area, they will have more collisions, therefore bouncing off of each other more and moving away. The random movements and unpredictable collisions of these beads represents diffusion.
Comparing this investigation with the microscope to the computational simulation, what do the particles represent in these 2 investigations? These models are a very simplified version of real-world diffusion. They may represent atoms, ions, or molecules that are diffusing to reach equilibrium, moving from a high concentration to a lower concentration. This can occur in anything from a biological process, spraying perfume, or mixing ingredients while cooking. The computational simulation would be most similar to molecules in air, such as the perfume example, while the microscope activity would be more comparable to diffusion within a solution, such as the biological processes or cooking examples.
The computational model shows the whole system of particles. In this activity, we only looked at a small subset of the particles. Therefore, the particles in the small area we looked at appeared to all be moving in the same general direction. The computational activity didn't take into account the changing velocity and acceleration of the particles. Their continuous and constant rate of motion is less realistic than the changing rate observed in the microscope activity.